Students, you are welcome to another lectures on chemistry of amino acids and proteins. If this is your first time of coming to my channel, kindly subscribe and share the content. So chemistry of proteins and amino acids. First and foremost, we are going to start with the amino acids, which are the building blocks of the protein. So we first need to understand what amino acids are before we now understand what proteins are made up of. So amino acids got their structure and classification, and we said that amino acids are the building units of proteins. So they are the building blocks that made of the protein. So there are about 300 amino acids that occur in nature, but only 20 of these amino acids are found in proteins, that is found in the biological system that are involved in many biological functions. So the structure of amino acid include 4 different groups that are attached to the alpha carbon. We have the amino group, we have the carboxylic acid functional group, and also the hydrogen atom, and also the R side chain. And this R side chain differs from one amino acid to another. So this is common to all the amino acids that are found in proteins. Means they have carbon, which is now bonded to hydrogen, bonded to carboxylic acid functional group, and also amino group. So it's the R side chain that differs between one amino acid and another. So for each amino acid, the side chain is different for each of the 20 amino acids. So at physiological pH, the carboxyl functional group is dissociated, and that leads to the formation of a negatively charged carboxylate ion, and also the amino group that is found on the amino acid is protonated, meaning it forms a positively charged ion, assuming a NH3+. And this now represent what we call a Zwitter ion. So if you look at the R side or side chain, and alpha carbon is bonded to an alpha amino group, which is protonated at physiological pH, and also the alpha carbon is bonded to hydrogen, and then the carboxylate ion, now the carboxyl group is now negatively charged. So this is what we now term as the Zwitter ion. So for proline is the only amino acid that is called amino acid. It is not called amino acid. So because the side chain is actually a different group that is formed. And if you look at the structure here, it has the carboxyl functional group and also this amino side chain. The side chain is fused to the amino group. So it is called an amino acid. So when we look at the classification of the amino acids, they can be classified by their R group. The R group being that it can be aliphatic R group, aromatic R group, or it can be basic or acidic R group. Yeah, and it can also be a neutral or a charged amino, polar or non-polar side chain. So for the chemical classification, we can also look at the chemical classification. We'll also look at the nutritional classification and also metabolic classification. So classification by R group, we can say that we are based on the polarity of the side chain, okay, or the charge of the side chain, and also we can define them in terms of their polar nature, those that are polar but not charged, and also those that are non-polar. So for the polar uncharged, they can also be polar positively charged. Okay, they can also be polar negatively charged or polar uncharged. Then based on the shape, we can say it either has an aromatic ring attached to it as the side chain, or we can have a straight line, a straight carboxyl, straight aliphatic group attached to it, which is a chain of carbon, hydrogen, okay? So we can also, based on shape, we can say it is now aliphatic or aromatic amino acids. So the classification according to the polarity of the side chain, we can have polar amino acid in which the R group contains a polar hydrophilic group. So it can form hydrogen bond with water. And in those amino acids, the R group may contain OH group, as in the case of serine, threonine, and tyrosine. Can have SH group, and which is thiol group, as in the case of cysteine. We can have amide group, as in the case of glutamine and asparagine. We can have amino group, as in the case of O-nitrogen, that act as a base. In case of basic amino acid like lysine, arginine, and histidine, we can also have a carboxyl functional group, as in the case of amino acid that are acidic, such as glutamic acid and aspartic acid. So the classification of polar uncharged R groups look as follows. We have serine, which now has the OH functional group in the side chain, which is the entire side chain is CH2OH. Now this OH now makes it polar. We have another side chain forming the threonine amino acid, and this is CH3, and it has OH, and also cysteine has methyl mercapto group, which is the thiol functional group, and CH2SH as the found in cysteine side chain. We can also have amide functional group, okay, as in the case of asparagine. These are the amino functional group, and also generally it's called the amide functional group, and we have it also as asparagine or in the glutamine. So positively charged R groups include lysine, arginine, and histidine, and these they can have a positively charged R group. We have the amino group here, fully protonated, positive here, also positive, okay? And these also can be classified as basic amino acid. So negatively charged R groups include the aspartates of aspartic acid or glutamates. And as we can see that the carboxyl functional group in the side chain of aspartic acid is negatively charged, so also in glutamic acid is negatively charged. And this, they can also be classified as acidic amino acid. So non-polar amino acid, the R or the alkyl hydrophobic group, it can't enter into hydrogen bond formation. There are 9 amino acids that are non-polar. We have glycine, alanine, valine, leucine, isoleucine, phenylalanine, tryptophan, proline, and methionine. So the non-polar that have aliphatic side chain, we have them here. We have glycine as the simplest amino acid, and it's achiral because the carbon atom here is containing 2 hydrogen, okay, and chiral carbon or chiral center is attached to 4 different groups. So as in the case of glycine, it is achiral. And here we have alanine chiral, okay, and proline here, we have it as amino acid. And we have branch chain here, as in the case of valine, leucine, and also as in the case of isoleucine. So the aromatic R groups includes those of phenylalanine, having phenylalanine here, the phenyl group, okay, phenol group or aromatic amino acid here. And we have the tyrosine here, okay, and we have the tryptophan, aromatic amino acids. These are the 3 aromatic amino acids. So 20 common amino acids are often referred to using the 3-letter abbreviations, and the structures, names, and abbreviations for the 20 common amino acids are shown below. So you should note that they are all alpha amino acids. So look at them. For the non-polar amino acid, non-polar aliphatic R group, we have glycine, alanine, valine, leucine, methionine, isoleucine. For the polar uncharged R group, we have the proline, serine, threonine, asparagine, glutamine, and cysteine. And also for the non-polar aromatic R group, we have phenylalanine, tyrosine, and tryptophan as the non-polar aromatic amino acid. We also have negatively charged R groups as in the case of aspartate and glutamate, and also positively charged R groups as in the case of lysine, arginine, and histidine. So each amino acid, aside from its name, has a 3-letter abbreviation and a 1-letter code. So each amino acid has the full name, their 3-letter abbreviation, and also 1-letter code. So for all the 20 common amino acid here listed, for amino acid glycine, its 3-letter code is GLY, gly, and 1-letter code is G. For alanine is ALA and A. Valine is VAL, V. For tryptophan is TRP, and W for phenylalanine is P, HE, and F, and serine, SER, and S. So for lysine, we have lysine, and also the 1-letter code as K. So these are the for all the 20 common amino acids. These are the representations of their 1-letter code and their 3-letter code with their full name. So here you know their structure based on their R side chain, okay? And here you know their names based on 3-letter notation and also 1-letter notation. So, and based on nutritional classification, we can have amino acids that are essential, semi-essential, or non-essential. So when we say essential amino acids, they are 10 in number, and they are ones amino acid that cannot be synthesized by the body. So they have to be taken in the diet. That is why they are called essential, and their deficiency affects growth, health, and also protein synthesis. While there are others that are called semi-essential that are formed in the body, but not in sufficient amount for the body requirement, especially in children. So arginine and histidine are called semi-essential amino acid, while the non-essential amino acid are those amino acids that the body can directly synthesize from either precursors or from other amino acids. Okay, so we have non-standard amino acids which are greater than 700 non-standard amino acids. They have been detected in living organisms. It means more than 700 non-standard amino acids have been detected in living organisms, and many are metabolic intermediates. For example, we have what we call ornithine and citrulline that are intermediate in the urea cycle, that is in the biosynthesis of urea. The biosynthesis during the formation of urine, before urine actually formed, that is what we call amino acid passing through the urea cycle. So ornithine and citrulline are also intermediates in the biosynthesis of the urea. We have amino acid derivatives, as we can say that chemical derivatives of amino acids are also having important biological functions. We have the catecholamines, and we have those that lack the alpha carboxylates of the amino acid. So the catecholamines, they lack the alpha carboxylate of amino acid. So we have the gamma aminobutyric acid, which is a neurotransmitter, histamine also, and also dopamine. We have GABA as what gamma aminobutyric acid, and dopamine as neurotransmitters, while histamine is part of the immune response. So function of the amino acid, apart from being the monomeric constituent of proteins and peptides, amino acid, they serve variety of other functions, some of which include amino acids that are converted to carbohydrates, which are called glucogenic amino acids. So amino acids that are converted to carbohydrates in the body are called glucogenic amino acids. So we have specific amino acids that give rise to specialized products. For example, tyrosine forms hormones such as thyroid hormone, T3, and T4. T3 is triiodothyronine, right? And T4 is triiodothyronine. And we have epinephrine and norepinephrine, which are catecholamines. And also we have the pigment that is responsible for our skin, which is the melanin. It's a protein that is formed in the skin and is responsible for the pigmentation of our skin. So tyrosine is now the precursor for the synthesis of all these amino acids. Now, also tryptophan can participate in the synthesis of a vitamin that is called niacin, okay? And also we have the glycine, arginine, and methionine that synthesize creatine. So we also have glycine and cysteine that helps in the synthesis of bile salts, and glutamate, cysteine, and glycine that are involved in the synthesis of glutathione, which is an antioxidant. So we also have histidine that changes to histamine on decarboxylation. We have serotonin that is formed from tryptophan, and glycine also is used for the synthesis of heme that is found in the hemoglobin, the protein component of the hemoglobin, which is now the heme. So we have pyrimidines and purines, which use several amino acids for their synthesis, such as the aspartate, glutamine for pyrimidines, and glycine, aspartic acid, glutamine, and serine for purine synthesis. And some amino acids also, such as glycine and cysteine, are used as detoxicants of specific substances. And methionine also acts as an active methionine in the process of S-adenosine methionine, and also the transfer of methyl group to various substances by transmethylation. There's also cysteine and methionine that are sources of sulfur in the body. So, and that is that about amino acid. Now let's quickly look at proteins. So for proteins, they provide the structure and also are part of what we call the cellular catalytic machinery, and they catalyze cellular reactions, and they carry other important tasks in the body. They have many biological functions, and they can be classified according to their biological functions in the body. So based on that, we can look at types of proteins and their functions. And also we have those proteins that are enzymatic. So most of them varied, and they have specialized proteins that are what serve as catalytic machinery in the body. And all chemical reactions of organic biomolecules in cells are catalyzed by enzymes. So enzymes are organic, you can say they are organic catalysts, they are biological catalysts that are found in so many reactions in the body that catalyzes so many reactions in the body. So we have other proteins that we can say they are responsible for transportation. They serve a transportation function in the body, call them transport proteins. They bind and carry molecules or ions to organs in the blood plasma. Like we have the plasma albumin, which is a transport protein in the blood plasma. We also have in this membrane, we also have aquaporins that is responsible for the transfer or transportation of water molecules. We also have lipoproteins that are in the blood plasma. They carry lipids, liver to other organs. So these are all examples of transport proteins. We have proteins that serve as nutrient and storage. And if we look at the seeds of many plants, it stores enormous nutrient proteins, and these proteins are required for the growth and the development of the germinating seedling. We also have ovalbumin, which is a major protein of the egg white, and also we have casein, which is the major protein of the milk that are examples of nutrient proteins. We have contractile or motile proteins, which are some proteins that are in the body. They are very numerous in the cells and organisms, which it helps the organism with the ability to contract or to change shape or to move about. We have tubulin as an example, which is the protein from which macrotubules are actually built up. So we also have structural proteins, which many proteins serve as supporting filaments, cables, or give rise to a biological structure or or protection. Okay, so it means they actually provide support. They serve as supporting filaments, cables, or shape. They give the protein shape, and it gives them biological structure and strength, or also protection to the organism. So a major component of tendons and cartilage is the fibrous protein, and we have collagen as an example, which is which is very high tensile strength protein. So we have ligaments that contain elastin, which is a structural protein also. And this structural protein can be found in so many parts of the body, especially the skin. So we have the defense proteins, which are now so many organisms defend against other, they use as defense system against invasion by other pathogens or also protect them. And we have immunoglobulins or antibodies, which are made by the lymphocytes of vertebrates, which can recognize and precipitate or neutralize invading bacteria. We have also fibrinogen and thrombin, which are blood clotting factors. Okay, they are responsible for preventing continual flow of blood when there is a cut in an individual. So those clotting factors help in preventing loss of blood. So we have regulatory proteins which help in regulating cellular or physiological activity, and the cellular response to many hormonal signals is actually mediated by a class of GTP binding proteins that are called G proteins. So they serve regulatory function. So we actually look at amino acids, which we call them the building blocks of the protein, and every amino acid has the same fundamental structure, which consists of a central carbon bonded to an amino group and a carboxyl group and also a hydrogen atom. So briefly, we have already looked at this. We just want to brush over it. And we have the side group, and there are 20 different amino acids. We remember that. And you can actually study more in the internet and look at their structures. Already we have looked at their structures and different side chains that are found in these 20 common amino acids. So try to remember and also assimilate as much as possible. So we have what we call a peptide bond, which is a covalent a bond that is formed during the process of building protein. And we have the polypeptide, which are polymers of amino acid linked together by peptide bonds. And these polypeptides actually made of the proteins. So with one amino acid joining to the carboxyl group of its neighboring amino acid. So this is actually how the protein is formed from one amino acid to another amino acid now actually joined to the elimination of water molecule to form a dipeptide, okay, via a peptide linkage. And also the peptide bond is formed when the hydroxyl group from one carboxyl group of the one of amino acid is removed and the hydrogen from the other amino acid is also removed. So the two OH and the hydrogen bond are now formed the water molecule that is now eliminated. So the formation of water molecule from the two amino acid is called dehydration synthesis. So it's this dehydration reaction that happens. So now the covalent linkage that is formed is known as the peptide bond, and also the molecule is now that is formed by the linking of the two amino acids is called a polypeptide. So proteins, they are very large molecules, and some protein consists of a single polypeptide chain. They have just one polypeptide chain, while others are multimeric, meaning they have multiple polypeptide chain, multi-subunits, and they have them in two or more. And we have individual polypeptide chain in a multi-subunit protein may be either identical or it may be a different. So if at least some are identical, the protein is sometimes called oligomers protein, and the subunit themselves are referred to as the protomers. So level of protein organization, we have 4 different levels of protein organization: the primary structure, secondary structure, tertiary, and the quaternary structure. So the primary structure here is depicted is the first level of the protein structure, and the second level is either the secondary structure, which is either divided into beta pleated sheet or alpha helix, which now forms the tertiary structure and eventually the quaternary structure. So the covalent bonds between the amino acid is normally defined by the sequence of the peptide bond, amino acid, and the location of the sulfide bond. That is what is in the primary structure, and the relative spatial arrangement of the linked amino acid is now actually unspecified. The now supercoiling of the primary amino acid, the coiling of the primary amino acid, and also the regular recurring arrangement in space of the adjacent amino acid residues in the polypeptide chain forms the secondary structure. And there are two secondary structure, most prominent secondary structure, which has the alpha helix and the beta pleated sheet. So for the tertiary structure, the now spatial relationship among all the amino acids in the polypeptide chain from the secondary structure gives rise to the complete three-dimensional structure of the polypeptide. That is what we term the tertiary structure. While for the quaternary structure, protein with several polypeptide chains has one or more level of structure that is referred to as a quaternary structure. So, and this refers to the special relationship of the polypeptides or the subunits within the protein. So, okay, I think we are going to stop here, and I can say thank you for listening and watching. If you're able to subscribe to my channel, kindly do so, and also click on notification bell so that you receive notification for our future contents. And if you have any question, you can post it in the comment section. With that, I say have a wonderful evening. Bye bye. See you next.